Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
Terminally ill patients often receive morphine to help them cope with severe pain. Yet chronic use of morphine and other opioids can backfire. Not only can the drug's effectiveness wane over time, but some patients find repeated doses make the pain feel even worse. Finding a way to block these long-term complications of morphine use would offer a major step forward in pain management, but the molecular mechanisms involved have been hard to unravel. Now, researchers led by Wen-Li at Fudan University in Shanghai have identified a cell signaling sequence involved in both the tolerance and increased pain sensitivity, hyperalgesia, brought on by regular morphine use in mice. Suppressing this pathway in the animals turned off both side effects, raising the prospect that the same could be done in people, the researchers report September 7 in Science Signaling. Both are glial cells, which support the nervous system. When the mice were exposed to morphine, these cells responded by increasing expression of a signaling protein called interleukin-33, IL-33, as well as its receptor, ST2. In the new study, the researchers found that through ST2, IL-33 activated the production and release of a cytokine called CXCL12 from the astrocytes. To test how the increased IL-33 and ST2 alter morphine's effects on the mice's pain response, the researchers used knockout mice that were missing the relevant genes, introduced inhibitory RNAs that wiped out the gene's expression in wild-type mice, or injected antibodies into wild-type mice to reduce. When these treated or modified animals were given morphine over time, they showed more reliable tolerance of pain than control mice, as indicated by behavioral tests that track when the animals move their paws and tails in response to heat. Collectively, these behavioral data demonstrated that spinal IL-33-ST2 signaling contributes to morphine-induced hyperalgesia, and analgesic tolerance, the authors conclude in the paper. They go on to suggest that injecting antibodies that target IL-33 or ST2 into a person's cerebrospinal fluid could be potentially used to prevent and/or treat the side effects caused by repeated opioid treatments. This study shows an important role of astrocytes in opioid-induced hyperalgesia. The findings are clinically relevant, he adds, but there is a long way to go. This study did not test clinically relevant drugs. Zakharou similarly cautions that, while the animals are a good model to explore the physiological response to morphine, mice cannot tell you they feel worse or that they have muscle aches. She adds that this is probably one of several pathways. Environmental and genetic factors could also contribute, as well as other molecular interactions beyond the spine. If the interventions in this particular pathway that the study identifies are sufficient, then we have a good target. But definitely there are additional and complementary pathways that one can think of. Quote, "Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you."